Morning, everyone. It's Friday for me. It's Thursday for you guys. Today's review day. This is the last bit of information that you're going to get before your test tomorrow. I'm going to be real quick today, so please do your best to keep up. That means, Tyler, get your notes down from up there where you store them in here and don't look at them at night. Let's make sure that we're going to get some major concepts together. I'm going to, as a matter of fact, real quick, I'm going to leave camera. I'm going to put my test in front of you so there's no questions about the short answer stuff. That way y'all aren't having any issues. You can still hear me talking maybe over here. So hopefully everybody is ready at least for tomorrow. If we're not, we're in deep trouble because we didn't do as well as we should have done on our sale. And it's not that y'all let me down. That's not it, right? I just know that y'all are more capable of that, and y'all do too. When we talked about questions, y'all had the answers. You just overthink them, and kind of like Bella said, your brain just turns to mush. So let's practice using some brains so that they kind of get worked out, kind of like muscles, right? So the more we think, the better we think later on. And so today, do that as we're going over these questions, kind of think about how I could ask these questions and know that there's going to be lots of which of these statements is false kind of things. And remember, all you're looking for in my test is literally the thing that I taught you. If I tell you 3 things that I didn't talk about, who cares? Doesn't matter if it sounds great or not. If I talked about one thing, if I talked about passive being no energy, and you see something in there that says something about passive being no energy, there's your answer, right? That's the kind of stuff that you're looking for. So just keep that in your mind. I am going to, before I finish the review type scenario, I am going to blatantly go over the end of the notes. There was not much left, so let me minimize myself real quick, because all we were really talking about at the end here was enzymes. So again, remember, we are pretty much giving the ability for us to turn something that had energy, in the sense of this case, we're breaking it down, okay? We played this video already, but we're breaking down to give us these products, so we're using the energy to do so, okay? And when we use an enzyme, we basically are going to lower that threshold, that activation energy threshold, so that we can create a quicker reaction, okay? Takes less stuff. So what fits into an enzyme is called a substrate, okay? And it's like a lock and key. And then I'll show you a video of this in just a second. It is a perfect fit. The enzyme's specific job is to break apart sucrose. The enzyme's specific job is to put together sucrose, whatever it is, okay? It's either going to be there for that purpose and that purpose only. Those substrates will fit in, and then it will allow the bond to be created or break the bond in either a hydrolysis, condensation reaction, whatever else it is. Okay, so I'll show you here. Here is our enzyme, right? And it's sucrase. Notice most of our enzymes end in ASE. Sugars again end in OSE. So this is an empty, right? It's available, okay? And so in comes our substrate, which is sucrose. It's bound together. You can see it here. What happens? Water comes in because why? Hydrolysis reaction breaks these apart, right? Sucrose. Here's our glucose and fructose, right? These two things, right, could literally come back and start the cycle again, okay? Once sucrose is here, and we can continue to put glucose and fructose off. Why do we want to do that? Well, because we like to break this joker down right here for ATP all day, all right? All right, so we have competitive and non-competitive inhibitors for an enzyme, okay? Feedback inhibition is going to be a massive role in metabolism. I'm going to go over this as you write this notes down. I'm going to give you just a second. A competitive inhibitor is literally another thing that bonds into the place of and why did I just literally just lose my mind there? I guess I'm thinking about hunting. The substrate, right? I have no idea what just happened to me. So our substrate that bonds, it's going to be a competitive inhibitor in which it literally bonds in its spot. Now I say bonds, it's going to go into that keyhole. If it covalently bonds there, you pretty much have a permanent inhibition, okay? And that can happen. Same thing with a non-competitive. A non-competitive inhibitor does not go into the actual keyhole. It actually joins itself to some portion of the enzyme, and it makes the keyhole useless. If you kind of get an idea of that, it alters the shape of the enzyme. So in other words, now the keyhole it's not the same. So key no fit. All right. So we're going to talk about feedback inhibition. You're going to have a question on my test about feedback inhibition. Specifically, it's going to be about the ideas of, can you explain how feedback inhibition affects metabolism? And I'll go over that here in just a second on one of these next few slides. Again, if y'all need to pause the video, y'all pause the video. So notice this: substrate, active site, right? Normal binding. Here we go. Competitive inhibitor, something's in the way. Here it comes, can't come in. Non-competitive inhibitor, it's something that changes the shape. Look how this looks. Now look how it looks right here. So it does not fit anymore, okay? Because it doesn't fit, end result is that the reaction can occur using that enzyme. Just that simple, okay? So this is what we call feedback inhibition, all right? And what that means is that here we are, we have a starting molecule, okay? And we have gone through the process of breaking down and breaking down. At some point, right, there is typically a feedback that one of the products will create an inhibition. Typically speaking, it will create a competitive inhibition in which it actually winds up inside here, okay? And that way it's in the way. Now, why would you want to do that? Well, let me ask you a question. Let's just say that this reaction started and this happened. And there was nothing necessarily that had to kickstart that reaction. It just wanted to go do it. And then let's say that this one happened and this one. What's going to keep the chain from stopping? The answer is nothing. So in other words, if we were breaking apart sucrose, we'd break apart sucrose until there was no sucrose to break apart. We'd break apart sucrose until there was so much glucose inside of the cell that the cell couldn't function and died. So instead what happens is that down the line, and it's different for different molecules, but down the line, we create a loop that says, stop doing this. It's kind of like, you eating at the buffet, right? I mean, you're down there at Sushi Village and you're going to town. And at some point, your body says, I shouldn't go any further. That's what happens with feedback inhibition. So on your test question, you're basically going to give me an idea of, we are feeding back, right? As enzyme is going through this process of breaking down things or creating whatever it is, there's going to be a feedback in that chain that literally says, stop making this reaction happen. Once we have used up the products, once we've used up, in this case, let's say B and C, and of course D has changed its shape. So once we've used up some of these and the cell knows that we need it again, then typically speaking, D will unbond, and as it does, the enzyme will go back to its shape and it may start that cycle again. And that could happen every day, every minute. It could happen every day, every few hours. It's all different for different things. So, but feedback inhibition is going to play a massive role in our metabolism, because if not, our cells would function so doggone fast that we just die. You couldn't put enough energy into them, right? You can't eat 6 of Bella's lunches and breaks and all the 47 things she brings each day. You couldn't do that because your cells would burn 5 million calories a day and you physically couldn't put it in your body. And it'd be because your cells were constantly at work. They have to be told to some degree, hey, let's not do this now, right? I got enough glucose. Can we wait? I mean, she's asleep. Don't really need to burn a lot of energy right now, let's say. Right? So why are you constantly creating more glucose? Feedback inhibition will give that ability. Okay. Enzyme inhibitors, right? Here's one that you all know, ibuprofen. Basically blocks pain receptors that are part of an enzyme. So we mess the enzyme up, so then the enzyme can't do that. Okay. Pesticides, another good example for those things. And then, of course, we've even used those in warfare. All right, I'm going to quickly go back to the beginning so that we can cover everything. Real quickly, since I have it in front of me, let's go over what is going to be short answer for my test, okay? Fluid mosaic model, you need to be able to describe it and tell me everything about it. Okay, why is it called that? We'll show that in a second. I need 5 types of proteins. Okay, I think I only gave you 6. So I want to know 5 and what their functions are. You'll see all these in these first slides. You cannot do really anything besides some memorization to put those in your brain. Listen, you can't skimp on that. I know you're like, ugh, I don't ever want to know that information. I'm probably going to never need it again. I got you. But you're talking about 10 points of your test. I mean, I got to believe that 9 of the 14 of you are not happy with a 90. You can't be. And that's assuming that you've got everything else right. So don't sell yourself short on that, okay? Just put that into your memory bank. You have time to do this. Rock and roll. You'll be fine. Active and passive transport, be able to differentiate. Not hard at all. Just give me a short statement about that. Active transport. We talked about 4 steps. Remember, on your thing, it shows 3, but I gave you the 4. All I want to know is the 4 steps of active transport. Endo and exocytoses. Okay, can you tell me the difference? And lastly, the feedback inhibition that I just gave you. Okay, so that's your short answer stuff. I just laid that out in front of you. You can play this video 9 million times if you need to, till you got what you need. Okay, so let's real quickly. Membrane structure and function. Fluid mosaic model, right? Diverse protein molecules suspended in a fluid phospholipid bilayer. In other words, fluid. The membrane itself is at constant motion, and this diverse protein molecules means they basically create a pattern, a mosaic. That's why it's called a fluid mosaic model. Okay? Our plasma membranes are selectively permeable. They allow things in and out. You should know what those things are. Those things are typically going to be small things, not things that are what? Polar, not things that are hydrophilic, right, which is the same thing, not things that have a charge. They love to let hydrophobic things in and out. They love to let teeny tiny things like oxygen, CO2, water, those things are no problems, okay? Here is your 6, they're all right here, okay? Enzymes, they can function as enzymes. They can function as attachment proteins, receptor proteins, channel proteins, active transport proteins, junction proteins, or glycoproteins. 1, 2, 3, 4, 5, 6, 7. I need 5. Okay? Don't care which ones you give me. However, I care that you are able to do this. What does a transport protein do? It's written right here. What does an enzyme do? Well, it speeds up reactions. What do attachments proteins do? Attach the extracellular matrix inside the cytoskeleton. What do receptor proteins do? Signal molecules bind to receptor proteins and basically send off inside something else. In other words, this thing for a receptor does not allow this item to come into here. It's basically saying, hey, this is touching this. This needs to happen down here, which we talked about like a doorbell. Okay? Junction proteins, intercellular junctions that attach adjacent cells. Glycoproteins, ID tags. Shorten the definitions. You got to give me more than just list 5 proteins. Tell me what the thing would do. Okay? That's all I'm asking you to do there. And should not be too complicated. Making sure I wrote and their function on there before one of you calls me out and says I didn't say that. Okay? All right, so if you feel free to watch any of the rest of these videos again, certainly can. Remember, this is the ideas of receptors and sending off information that's going to happen, all because of signal proteins. You can see them there, signal protein. Again, whatever is coming into the cell is not what's binding here, okay? It is something else. It's a signal. It's something on the outside, and it's extracellular matrices, just like that, okay? Evolution, we can care less about because it doesn't happen, right? All right, diffusion. Tendency of particles to spread out evenly in available space. Goes from down a concentration gradient. Goes from high to low. Chooses to get into a equilibrium. In other words, a balance between what's on the outside and inside. None of these questions should be things in which you're looking at me going, I don't understand. That's what diffusion is, okay? Because it does not require energy, what do we call it? Passive transport, okay? What things can we passively transport? Water, O2, CO2, okay? Those things should be easy to see. Here we're looking for equilibrium, right? Don't miss these questions, all right? Do not miss these questions. Membrane selectivity. In other words, it wants to allow things in and out, but it's specific things, okay? If we are diffusing water, we call that osmosis. If we are diffusing water, we call that osmosis. Osmosis is the movement of water from what? And don't tell me high to low concentrations. It's from what? Low to high concentrations? Okay, of what? Low concentrations of solute to high concentrations of solute. Don't not know that. You got me? Don't do that to yourself. It is low concentrations of solute to high concentrations of solute. We talked about that. I described that on the board very, very well, okay? This idea that we are going to add water to something that is high in concentration so that it will lower it, so that what we are taking the water away from will actually go up in concentration and balance those two things out, okay? That's osmosis. Again, remember the reason why we do that is because water is easy to move and doesn't require energy. Cells want to be lazy, so do we, okay? You saw and you remember this question, right? Predict a net water movement between two solutions, 0.5 sucrose solution and 2% sucrose solution separated by membrane. Water is going to move from where to where? From low concentration to high concentration, okay? Technically, it's moving from high to low concentrations of itself, of water. There's higher concentrations of water here than here, okay? So it's still passive, but it is moving from low concentrations of solute to high concentrations of solute, okay? You stick it up here. Think about what that also means. Now you know three other things that go along with that, right? Do you not? Hypertonic, hypotonic, and isotoners. Yeah, that's right. If you happen to remember watching Ace Ventura: Pet Detective. Here they are. Hypertonic solution. Higher concentration outside than inside. Water moves from low to high solute concentrations. Water leaves. Hypotonic. Higher on the inside. From low to high, water enters. Isotonic, they're equal on both sides. If water leaves a cell, it shrinks, both in an animal and a plant. If water enters a cell, it comes into and probably lyses an animal cell. Okay, cytolysis, what we call this. This over here happens to be a very happy plant cell. What is this thing called? Starts with a T, turgor pressure. Isotonic, perfectly happy animal cell, and we prefer isotonic solutions. Over here, where I messed up my drawing because I think I just put my hand on it, over here, what do we have? We have a flaccid or a limp, right? That's what we mean by this. Limp cell, that's a plant. It's not happy. It needs that turgor pressure and that cell wall to give it enough structure, okay? Doesn't have it. We shrunk the vacuole down, okay? You better know these concepts. Guarantee you they're on your test, probably 7 different ways, okay? Got to know them. Whoops, I just tried to move to the next slide. And it was just my beautiful face. Good example here, right? We wound up with more water over here. I understand that. But guess what? Our solute concentrations are probably the same. We have way more solute and way more water. We have less solute and less water. Concentrations are more equal. Okay? All right, remember, tonicity is the desire of a cell to gain and lose water. Here's the hypertonic, hypotonic, and isotonics. Went over all that. Should not be wondering what I'm talking about there. That should be very straightforward, okay? Hypertonic, hypotonic, and isotonic, okay? Here's a good example of what happens. Remember what we talked about, right? Cell lysis is called cytolysis. When we shrivel up, remember this is called plasmolysis for a plant, okay? You can see that right there. Plasmolysed, see they're shriveled on the plant membrane, bottom right. Okay. And then here, you have something called crenation. It's what happens for an animal cell. Crenation. Crenation. We looked at all these, right? Watch what happened. Hydrophobic substances, easily diffuse. I mean, I've already said it, so you know it's going to be there, okay? Polar charged substances do not easily cross the cell membrane. You know it's going to be there, okay? However, we can move polar and charged substances in through what? Facilitated diffusion. Sometimes. It depends what they are. Okay, glucose would be a really good example. Water, would be a really good example. Aquaporin would be a good thing that we could talk about for facilitated diffusion. Again, does not require energy. You can see it right there. And notice it says relies on a concentration gradient. What does that mean? Goes from what? High to low. That's all that means. Okay, it's going to go from high to low concentration. That's has to happen if we're going with the concentration gradient. The only other way would be against the concentration gradient. We're not going against the concentration gradient unless we have what type of transport. That's right. It's active. It's not passive. It never will be. Okay, stick that up here. All right. Active always requires energy. All right, transport proteins help specific substances diffuse across the membrane. Very rapid diffusion of water is called aquaporin. We bring glucose in because glucose can be free for us to bring in. No ATP used to do so. We break glucose down to turn it into ATP. That sounds like a great plan. Okay, that's like me giving you free money to make money. Okay, that's a lot better than you having to have your own money to do it. So let's keep that stuck up here and kind of keep our minds going with what it's supposed to be. Remember, they're typically very specific. They only allow certain things in. You can see the shape of this channel is specific to this round shape in this case. They're obviously not typically going to be this perfect, but it does not allow this in. At the same time, others will allow this in, okay, like a lock and key to some degree to let them pass in because they're not overly heavily regulated in the sense of trying to keep this protein free of anything else that's not supposed to be there. So they just try to block them from coming in to begin with, okay? Active transport. Must expend energy, okay? Must expend energy. A energy molecule, right, ATP, is going to be that energy. You are going to need to know on a picture what ATP looks like, and you're going to need to know how we create energy from ATP. In other words, what happens to it chemically? What is changed about ATP that then releases energy? You'll see that on a picture in a second, okay? All right. 4 stages of active transport. I told you what these were. Remember, solute binds, ATP changes the shape of the protein. Remember, number 3, solute leaves. Number 4, protein returns shape. I've been over this. These are 4 things. These are 8 points of a test. That, along with your 5 proteins, okay, is what, 18 points of your test. Best you can make is an 82. Seeing as how we do on some of these multiple choice questions, I think it's now time to do well here, okay? Solute binds, protein changes shape because of ATP. Make sure you include this. Protein can't change shape because it just wants to, okay? ATP can bond and then release, and when it releases, this can happen, okay? So ATP bonds, shape of the protein changes, solute leaves, step 3, solute 4, or so not solute, step 4, technically, protein will return to its shape, typically because the phosphate's going to let go of right here, which is what we see exactly when we look at our sodium-potassium pump, which is right here, okay? Sodium-potassium pump, you need to know some ideas about that. Sodium is going to do what? Sodium going in or out? Sodium's going out, right? How many are leaving? 3. What's coming in? Potassiums, how many are coming in? 2. It is a 3 for 2, okay? Membrane potential is what we call the difference between this charge and this charge. We're going to have 3 positives leave and 2 positives come in. You can tell there's a change in positives. We have a net gain in the extracellular and a net loss here, okay, in this case. Again, you can watch all these videos. By the way, all of these things now are on your hindsight. I'm going to try to tell you that today in class if I get to see all of you. But I put the actual PowerPoint up in Heinz's thing. I could not do it any other way. I am going to make sure that you know the links to these videos in case you want to watch them again. They'll all be on YouTube, okay? Because I couldn't figure out any other way to upload them. All right, so subscribe to my channel, everybody. I'll be a millionaire one day. All right. You're going to give me the definition, the differentiation of these 2 things in my test, exo and endocytosis. Exocytosis is what? Process of bringing things out. Endocytosis is bringing things in. Can you already tell me some things about them? Blakeney Bird, endocytosis. Tell me about them. Don't tell me just bringing things in. What kind of things can you talk about? Are there other names for this? Okay, so we have phagocytoses, the bringing in of food particles. We have penocytoses, the bringing in of fluids, right? Feeding process, drinking process of the cell. And lastly, we have receptor-mediated endocytoses, meaning we're typically bringing in solutes because receptors on the outside of the cell have bonded to them, have bonded to the solute and says, ooh, now it's time to bring this into the cell. Which of these things, CADE, results in an increase in cell size of the membrane? Exocytoses. Remember, endocytoses, we're using our own membrane to pinch and then to come in. Exocytoses, we're taking something from the Golgi apparatus, let's say, that's been shipped to it, and then that thing is now bonded with the actual membrane. As it does that, what do you wind up with? You wind up with more phospholipids there, okay? So Should be pretty easy to see that, right? Here's phagocytoses, engulfment of a particle, typically food, okay? Receptor-mediated endocytoses, use of the membrane receptors for specific solutes. Again, same thing, just told you that. Look, here's the question that I just asked Cade. Does this involve endo or exocytoses? Should not be difficult. Here we are, right? Food vacuole, right? Phagocytoses, here we are. Look at this. Here's our solutes that we brought in. Here's our receptors that pick these things out, okay? Gave us very simply a receptor-mediated endocytoses. Exo there, right? So that's the leaving of those products. Pino, the bringing in, again, of liquid or fluid, all right, from the outside. Typically, solutes will come with it, but we're bringing in extracellular fluid as we do so. Phagocytoses, we're going to be bringing in this solid piece of food, typically, okay, is what normally it's going to be, but at least typically some solid product. Receptor-mediated, we're looking to bring in this joker, right, this joker, whatever those things may be, these solutes, because we need them. They can bind to these receptor protein. Remember, the receptor protein is a protein, okay? Test question. What would, in receptor-mediated endocytoses, right, I would say this drug, right, is part of receptor-mediated endocytoses. The receptor is made of what, or is a what, that will bind and allow this solute to come into the cell? It's a protein, right? You know this because it's built into our membrane. Our membranes are made of phospholipids and proteins. It's basically it, okay? This protein would bind to this, bring it into the cell simply as this pseudopod and this pseudopod reaches around, right? Pseudopodium, okay? Energy. Remember, we talked about all of this mess, and there's nothing to do besides get your head wrapped around the physics of this. Kinetic energy is something moving. Potential energy is the stored energy, right? And can include chemical energy. Need to know your laws of thermodynamics. They're going to show up on your test. Energy can change form but cannot be created or destroyed. And as energy transfers or transforms into other forms of energy, typically there's going to be disorder, right? And what we mean by that is that there's not a complete exchange. The same amount of energy existed, right? In other words, there is heat put off. It's still energy, okay? It's just not that all the energy went from point A to point B. Some of the energy went from point A to point B. Some of it was lost as heat energy. That's what happens. That's called entropy, disorder, okay? So you're just going to have to know it because that's important for our cells, and I'll show you that in a second, all right? Remember, cells act pretty much like a car. Take some kind of fuel, glucose and oxygen, just like gas and oxygen. We have combustion that happens through cellular respiration. Heat is put off. ATP is created for the cell. Carbon dioxide and water is a byproduct. Carbon dioxide and water is a byproduct here too. This creates movement or kinetic energy, right, of wheels. I need to show you this here. Whoops. Right? Creates kinetic energy of wheels. This creates kinetic energy of what? Movements of cells. Perhaps it's muscle fibers that are going to contract. That's why when you get through running, if you were to touch your skin, touch your muscles, everything's warmer. There's a reason, right? Energy has to be given off. Why? Because we've been changing energy from one form to another. So there's been some entropy. There has been some disorder. So some heat is lost. Please watch this video again. I cannot harp on this enough. I think this video is key for you knowing this information, okay? Because they do a very good job of going over the ideas of physics far better than I can. If you have any questions about this, step down the hall and go see Roos, okay? For sure. Exergonic reactions. Exo, releasing of, exit, releasing of energy, and exergonic. Endergonic require energy. They yield products rich in potential energy. In other words, we build them up for the future. It's kind of like, well, I'm not picking at Bella again, but it's kind of like Bella's lunchbox, okay? It's an endergonic reaction. She is storing up for the future, right? Exergonic is her using that food to then give herself energy. But endergonic would be her storing it, her putting it in there going, you know what, this is going to give me something later on. It will, okay? And metabolism encompasses all of the cell's chemical reactions. In other words, the control of everything that is both exer and endergonic is what we define as metabolism, okay? Here is, right, our potential energy of molecules, okay? So in this case, our reactants, right, had lots of energy to give off. As we turned them into products, the energy was released, right? That's what we've got right here. This is the difference. We also have this. This is the building up. So in this case, let's go back and look. In this case, we have a what? An exergonic reaction, right? In this case, we have an endergonic reaction. Why? What have we done? We have put energy in, and now what we have left over, right? Right here. What we have left over is giving us the ability to then use those products in the future to burn this back off, okay? So endergonic here, exergonic as we put off here, okay? ATP, adenosine triphosphate, powers all forms of cellular work. Notice this, the transfer of a phosphate group from ATP is involved in chemical transport and mechanical work, okay? Know the picture. Hint, 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 hint. Know the picture, okay? Here's your chemical work, right? Phosphate's taken off, making ADP. Transport work, phosphate does what? Bonds here, opens this up, solutes leave. When the phosphate leaves, this will close back down, okay? Very simplistic. Mechanical work, specifically right here with muscle fibers, right? Our motor proteins helping to move these things. ATP is involved in everything that happens, right? Inside of a cell. Here is basically the loop, right? ATP, we're going to break it down, going to use that energy that's put off, got to put it back together. Cellular respiration is going to help us do so. Off we go, we've got ATP, we do it over and over and over and over and over, and your body does it all day long, okay? Which is why you're so tired when you're in my class sometimes, right? Enzymes, just about done. Again, they're catalysts. What do? They don't speed up a reaction. They do speed the reaction up. But how do they do that specifically? They lower the activation energy, okay? Here we are. Activation barrier takes this much energy to make this happen. Enzyme pushes us down. Only takes this much energy to make this happen now, okay? We like that. It's less work that our bodies have got to do, okay? That's all that's telling us. Lowers the activation energy barrier, okay? You can see those pictures right there, right? So what it took to make the reaction happen, without it, this is what took with it. Far less energy required, right? Far less energy. And this is the energy that we probably either stored, right, from reactants to products, or that we gave off, right? The change of energy from reactants to product is what C is here, okay? So we were either putting in or giving off this much of a difference of energy. Either creating an exergonic or an endergonic reaction is all that means, okay? That's what this question's asking you there, okay? Again, here's our enzyme. Basically, what's going to happen is we're going to lower this threshold so there's less of a gap is all that's going to happen. Again, please feel free to watch that video. It's perfectly fine, okay? That's it, because I can't go over anything else because I've already gone over the rest of this. Substrates and active sites, competitive and non-competitive inhibitions. There's your active sites, glucose, fructose, substrates, entering into those things. Got to know the names of them, okay? Substrate is a big key. Enzyme shape is important. Did I ever say anything to y'all about how you could denature proteins? That's right. Temperature, pH. Is an enzyme a protein? So would there be a way that I could affect a enzyme so that it wouldn't do what it's supposed to do? What would happen if I heated it up or changed the pH? It's going to change its three-dimensional shape. If the keyhole don't fit the key, can't do anything. Okay? Competitive, non-competitive inhibitor, feedback inhibition. Remember, feedback inhibition is a discussion question. The ideas of competitive and non-competitive inhibitors are very simplistic. Okay? Competitive is in the way. Non-competitive here, right? Basically changes the shape. Which of these things is typically going to last longer? This. This thing can probably fall back out of here. This thing is bonded and changed the shape of this enzyme. Can this unbond? Yes, it can. It's not as common. If you see a covalent bond of this, it's done. Basically, it's not going to let go. Shape's going to stay different forever, and this enzyme is no longer functional. Cell will typically discover that and find a way to go bring a lysosome to engulf this thing and to start using something to break it down. It's basically saying this protein, which is an enzyme, is useless. It's not doing us any good. Remember your feedback inhibition. Should be no issues there. Okay. And there you go. Here's the things you should be able to do. Here's your active and passive transports, right? Down concentration gradients, across concentration gradients. I'm not going to have to draw this picture for me, but you should understand everything that's happening right here, right? Memorize these charts. Just kidding. And that's it. Okay. You survived it. And you're going to survive your test tomorrow. Okay? I'll be back on Monday, if I'm not already back, and get to grade papers for like 4 days as I try to catch up and be excited about seeing y'all again, right? I will tell this class, because this is probably the only class that I'll be able to tell. I don't have much planned for the Monday that I'm back. It's going to be a try to figure it out day, at least initially for me. Because I can't think that far in advance, apparently. I don't know. Just mine's not there. So maybe today, during my half day, I can figure out what I'm going to do with all my classes when I first get back from my trip. Hopefully, I go and I'm there for like 4 days and I'm back. And so I can come back during the week that I've already got planned, and then I can figure things out. All right. This video is 36 minutes long, 37 minutes long. Hope you guys do well. I really do. I've been proud of y'all's work so far. I've really enjoyed being with you guys. And so let's keep working. Okay. It's homecoming week today, I guess, technically. And this advice is not going to help you for Friday. So I'll just go ahead and tell you today during class, but I just want y'all to understand. I want y'all to be the people who you want to be known as. Everything you do. I think most of you are smart enough now to know that being the cool person doesn't matter. So do the right thing. Wherever you are. Kenan, shut the door. I'm videoing. Read the sign. I am giving you the ability to, sorry, Kenan decided to come in and say hello to us. I want you to do all that you can to be willing to put the right thing first and to say that you know better. Whatever that is. It doesn't matter what it is. I don't care. I'm just saying, because y'all are to this point where you've discovered no one's going to remember you because you're cool and awesome. They're going to remember you because you either did the right or the wrong thing. So when it comes to class, when it comes to dating relationships, when it comes to how you treat parents and teachers and whatever else, just be the better person. I assure you that when you meet for your 5 and 10 year reunion or whatever else it is, there is a 0% chance, and I can say this as honestly as I can, there is a 0% chance that anybody remembers you because you happen to be the wild person and they thought that's cool now. They won't. They certainly will remember you because you stood up for what was right in high school, and they made fun of you, let's say, but they remembered you for it, and it impacted them today, right? So just be good, be great. If I get a chance to see you today, I'm going to ask you to do the same thing for homecoming Friday night and to be the person that you should be, okay? So look forward to being back with you. Thank you very much. Take care, and bye-bye now.